A very good afternoon to the young NEET aspirants. I keep saying this because achieving something at a younger generation is very good, and especially NEET or any entrance exam. So Vanakkam, Namaskar. We would be happy to see that if you come up with comments like hi, good afternoon, so that I'll get to know that how many of you have been seeing me live today. Okay. So, yes, my friends, you have been there with me from past 3 classes. We have been dealing with respiration in plants, and this is the climax class, I could say, the climax thing of the chapter. Like you say, before rating a movie, we generally see that what is the climax of the movie. That is what we ask our friends, did you watch that movie? What is the climax of that movie? So based on that, we rate it actually. So to rate respiration in plants, which is got beautifully designed inside a cell, we should know what is the output of it. So much the cell has undergone, so many cycle has happened, so many enzymes have been involved in. How many molecules of ATP is out from aerobic respiration? So that's how we are going to see the climax of this chapter, and then we are going to rate out respiration based on the number of ATP released. And one more thing, we are going to study about RQ factor also. That's called as respiratory quotient. Okay. Yes, my friends, we studied about ETS, the last step of aerobic respiration. Okay, we talked about the chain, how beautifully it's got designed inside the inner mitochondrial membrane. Then we studied about oxidative phosphorylation. That's nothing but the ATP synthase, how chemiosmosis is maintained in that, so that on driving force of protons into this ATP synthase, ATP is got produced from ADP and the molecule of inorganic phosphate. We studied overall ETC in the sense how many ATP molecules are produced from one molecule of NADH and how many ATP from one molecule of FADH2. This is what we studied. And one more important thing which I noted down is every coenzyme, each coenzyme requires one atom of oxygen. Okay. Now, one more thing which we are going to deduce in this class is how 6 molecules of oxygen is being utilized in overall equation of respiration. Okay, we discussed about how 6 molecules of carbon dioxide is out through Krebs cycle and link reaction. We did that in the previous class. The question which has been gone wrong by many of the students in our CPP, by our passport users, I see most of them have answered option C for this question. Let me read out the question once again. The complex with cytochromes B and C1 or BC1 is, it is cytochrome reductase. I think आपको कुछ problem हो रहा होगा उन complex के नाम को याद करने पर, or if you have you haven't understood how the chain is been organized and how it is undergoing stepwise electron transfer. I'll in detail, if you want previous class, please go in there. I have described it neatly. Okay, that we'll talk about it later in this class also. Okay, to stick with me with this CPP question, what has gone wrong is in the sense, now complex 1, I'll just write it down here so that it will be easy for you to understand, is your NADH, write in a short form, okay, reductase. This is complex 1. Complex 2, okay kids, you have put the answer as succinic dehydrogenase. That's wrong, because I'm asking cytochromes BC1. This is an enzyme, it's not a cytochrome complex. So complex 2 is gonna be your succinic dehydrogenase. Okay, I'll show you the picture what we discussed in the previous class. Okay, these both given there, protons and electrons, from here FADH2 comes in, from complex 1 NADH plus H plus comes in, this gets inside ubiquinone. From ubiquinone, okay, it goes to complex 3. Complex 3 is nothing but, what is I'm asking, the complex with BC1 is your complex 3. What is the name of that? Okay. Okay, it's not there in the option actually. It is called as cytochrome C reductase. This is your complex. Okay, I'm sorry, it's the option 1. Okay, it's called QH2 cytochrome C reductase. It is the option 1. So even I get confused with this. To keep it memorized completely, okay, Give it a shot every day or once in a week. Write it in the paper. Okay, I know it's a little bit confusing, but nothing is impossible. You can make this possible. To make it fix inside your brain is keep writing it. Okay, it will get memorized to you. So this cytochrome C reductase becomes your complex 3, where there is B and C1. Two cytochromes are there, two molecules are there. We discussed this. So from complex 3 it goes to cytochrome C, your mobile electron carrier. From cytochrome C it goes to your complex 4. Okay, complex 4 is the one which is called as cytochrome C oxidase, which has two molecules A and A3. This is what we discussed. Okay, so look at the questions. You have two clues actually. Which one has the cytochromes BC1? So that will be obviously your cytochrome complexes. The answer is succinic dehydrogenase. It's an enzyme. There is no cytochromes in there. BC1 is actually there in complex 3. That is cytochrome C reductase. AA3 is in cytochrome C oxidase. I hope you have understood this. Any problem? Anything which you haven't understood? Please raise it through the comments, please. Goals of the class. We keep repeating this actually, isn't it? So enthusiastically, we are all the viewers of this movie called as Respiration in Plants. So we are going to judge it, we are going to give the rating to it by studying, what is it? Respiration balance sheet. Okay, I am just putting the tick mark before we have completed studying it, but just to put a tick mark on what we are going to study in this class. Then respiratory quotient, it's called as RQ. We'll be telling this as RQ throughout the class to consume energy and ATP, not to put a lot of energy in saying respiratory quotient actually. We'll keep saying RQ throughout a class. The clever cell. What am I talking here? Okay, the clever cell. As you've been studying photosynthesis, respiration, biomolecules, so many chemical reactions inside the cell, can you notice one thing? The cell has been hard working so much to form the compounds in terms of the circumstance, that you mean to say in terms of the surroundings. Now I'll give you an example. In your school or college, when you would have seen, your teachers will set the timetable in that way, so that in the morning half, when you're too attentive, the subjects which are complicated will come into picture, okay, like math or social, things like that, so that you'll be attentive to listen. After your lunch, there are chances, there is a human nature that we doze off, we tend to sleep off, okay. So at that time, the chapters is lightweighted, will be kept post lunch. Please notice this is how your timetable has been designed of, so that to keep you awake throughout the school hours or college hours. So what and all, our principals and teachers have to do to keep us awake and attentive. Now this is what the cell is also doing. What is it doing? Okay, in plant cell, we know that through photosynthesis, carbohydrates is synthesized. But you know the story that it is getting stored in the form of starch. Why is it actually? Starch is much more insoluble in water. It does not get solubilized in water. Now to keep the osmotic potential balanced with the plant system, carbohydrates has been stored as starch. Good thing, no? See what and all it's been doing. And during photosynthesis, the highest energy is been trapped in carbohydrates, because it's easy to get broken away. Okay, so that is why it's been kept as carbohydrate, synthesized as carbohydrate, stored as starch. Okay, here energy is stored in carbohydrate. It is stored as starch because starch is insoluble. And when we have studied glycolysis, that when carbohydrates get inside, respiration gets inside glycolysis, it is sucrose which is getting converted into glucose and fructose. Okay, why is this happening? Because sucrose, fructose and glucose are much more solubilized in the cell. So during respiration, from the starch, this all carbohydrates has to move to many cells, okay, when they have to get respired. That is translocation of solutes. Now why sucrose is getting transported? It is only sucrose which is getting transported because it is less reactive when we compare it to glucose. See, getting deeper inside these concepts is out of our discussion point here, but we are just jotting out the points to see that how cleverly our beautiful cell is getting mechanized based on the circumstance. If it has to get transported, it's converting into sucrose. If it has to get respired, it's converting into your fructose and glucose. If it has to get stored, it is in the form of starch. If it has to get synthesized, it is in the form of carbohydrate, that's mostly in terms of glucose. Okay, so many things it's doing, so much of circus it is doing. Okay, I've put a picture over my head. Okay, this is what the cell is announcing, saying that I'm clever enough to maintain the metabolism, inside any living organism, let it be a plant or an animal. We'll talk about an animal cell. So what and all we eat, okay, we actually store it. The energy is getting stored in the form of fats. Majority of it is getting stored in the form of fats. This is the fats which is getting inside our Krebs cycle. We saw that an amphibolic pathway actually. Other than if it is carbohydrates, how fats gets inside respiration. Here are beautiful fats, as we saw it in the previous class. I'll keep repeating it so that it gets rejuvenated. There are a lot of energy getting rejuvenated and things, it gets memorized into your brain. You start picturing it. Okay, so in amphibolic pathway, we saw fats getting converted into fatty acid and glycerol. Fatty acid got inside acetyl-CoA. It became into acetyl-CoA. It entered Krebs cycle. So our fats actually enters Krebs cycle in our body. Now when we exercise, it is the fats which we are burning off. Highest energy stored in it is getting formed into form of adipose tissue. We have to burn the adipose tissue. I keep talking about gymming also. We talked about fermentation, the lactic acid fermentation also. Look at it, how beautifully it's got mechanized, and that's why you should say the clever cell. Now how cleverly this cell is being beautifully using its brain and doing all these things. Obviously the end output, I mean to say the ATP synthesis should be that valuable for the cell. For it's going through so much of circus, the output should be that valuable for the cell, so that it feels I've done so much, okay, because I've got so much of ATP out from aerobic respiration. I went through glycolysis, I went through Krebs cycle, then at the end I got so much of ATP. Isn't it rewarding of? You have to reward the cell and the process. Yes kids, we are going to start studying respiratory quotient. I said we'll be saying it as RQ throughout this class. What is a quotient? I'm not going to write off. If you know about it, please put it on comment. If not, I'm there here, I'll help you out. A quotient is called as a result or an amount obtained by dividing a quantity by another quantity. Okay, this is what a quotient is called. If you want to know much more about it, Google it, you'll find it out. Okay, this is just to start off what is a quotient. RQ quotient in plants is actually, you can see on the screen, is given as volume of carbon dioxide evolved divided by volume of oxygen consumed. So in aerobic respiration, oxygen in, carbon dioxide out. So how much of amount was out divided by how much of amount was in, that is carbon dioxide out, oxygen in. Why are we doing this? I said quotient means we are dividing something to get a value. The value, based on the value what we get it as RQ, based on it we are going to tell whether the respiratory substrate, okay, or the one which went input inside respiration, the molecule, whether it's carbohydrate, fats, proteins or organic acid. Okay, we are going to help out the plant. Okay, no, actually we are helping ourselves by knowing what is the plant actually oxidizing. Is it always carbohydrate? So based on this RQ value we are going to predict now. Okay, so before we predict it, I'll just give you the values what and all you get for, as I have given here, respiratory substrate. Let me put it in a different color. It will look much more, the picture will be clear and be interesting. The RQ value for carb, I have just put the short form, it's carbohydrates, is 1. Just right side, when you scroll up, I scroll up the pen to the right side under organic acid, it is more than 1. Interesting, how? We'll discuss about that. Protein, it is at the level, it's at the range of 0.8 to 0.9. Fats, the least RQ level, 0.7. We are going to discuss this. 1 is our carbs, extreme level organic acid, Some were doing justice with 1, 0 and 1 is our protein, 0.8 to 0.9, the range. Fats is the low one, 0.7. How did they get this? Let's discuss that slide by slide. So, I have just made a small table out here so that it will help you understand in what such condition, what respiratory substrates are oxidized and why the values are 1 or more than 1, less than 1. Why? Actually, I have given the reason also. As you note the table which I have put here. So, the type of substrate I am talking here is carbohydrates. I told you the RQ value is 1. The reason, because you see this reaction, this is the overall reaction of respiration. We said RQ value is volume of carbon dioxide evolved by oxygen taken in. So, how much is evolved? 6 carbon dioxide. How much is taken in? 6 molecules of oxygen. This will give you 1. Okay. Why it is 1? If anybody asks you why the RQ value of carbohydrates is 1, because the volume of oxygen taken in and carbon dioxide given out is actually equal. That is why. So, where are the plants or where are the conditions where carbohydrates has to get burned out as a respiratory substrate? This will happen in places which are undergoing rapid growth. Okay. So, that you can see it in germination of seeds like cereals and tubers. I have given the examples at the a straight stretch on the right side under condition of plant. Okay. I hope you have understood this, how it is 1 and what condition it takes place. Let's jump into the next respiratory substrates, my friends. Fats. I told it is less than 1. It actually ranges at 0.7. So, this equation may look much more complicated for you. You have to, I would not say that you have to completely memorize this, but keep noting down how many molecules of oxygen and carbon dioxide is taken in and evolved respectively. Okay. So, this is tripalmitin, one of your fats. Tripalmitin, it's a fatty acid, palmitin. Okay. It has a complicated, the composition. It is C51. Okay, and let me not write it, it's actually written there. Okay. Use the green pen. So, it is C51H9806. It is utilizing, oh my god, 145 molecules of oxygen. Why so much is because, so we are talking about the types of substrate fats. We told it is less than 1 because chemically fats have low oxygen into them. Okay. The composition of oxygen is very poor. Hence, they need more molecules of oxygen to oxidize them. That is why it is 145 oxygen. The amount of carbon dioxide released is 102. Okay, and water is 98 molecules of water. Just keep a keen concentration of how much of carbon dioxide released, my friends, and how much taken in. Let's come to the RQ value. What we are supposed to do? 102 divided by 145. You can use your calculator also here. I did that previously. Okay. So, the value becomes 0.7. It's much more less than 1, right? Why? We told the reason also now. Where can you see this happening is where the reserve food material is fats. It's oil. Okay. You can think about the seeds. Sarson ka tel, aapka mustard, castor seed, from which all our cooking oils comes, those oil from which we light the lamps and all. Okay. So, this is actually seen in germination of oily seeds where the reserve food material is highly fats. Mustard, castor seed can be the example where the respiratory substrate is fats. Okay, kids. Next, we are left with 2 more respiratory substrate. What is that? Organic acid and proteins. Organic acid, as I said, is more than 1. Now, to picturize this, where do you think the reserve food or the reserved compound can be? Malic acid. Malic acid is one type of organic acid. Where do you think you can find this? You must have all studied about CAMP pathway, right? Your crassulacean acid metabolism, the CAMP pathway. There, to reduce transpiration and to take over photosynthesis, our beautiful cell, it stores off the one which has to get burned off, okay, your carbohydrates, in the form of malic acid. You must have studied that. So, this kind of respiration where malic acid is getting respired, the situation can come up is within succulents. Succulents are the place where you can see about CAMP pathway, you have studied about it. So, when malic acid is respired, the RQ value, no need to know of any equation here, it is out of the scope, not required. So, the RQ value is more than 1 because these organic acid are rich in oxygen composition, okay, shows here they are rich in oxygen composition. That is why very less oxygen is required for oxidation. That is why where there is the oxygen level of requirement is low, always the RQ level shifts more than 1 or from 1 to more than 1 it shifts off. So, again the next respiratory substrate which we are going to talk here is proteins. See, this is all memory based kind of concepts, but you have to appreciate it. As I said, the clever cell, what and all it is doing to actually match up with the circumstances. What is the circumstance here? Which condition it can happen when the cell has to go to the protein and ask for a let's oxidize you, man, we have to release energy now, is under severe starvation. When the plant is undergoing severe starvation, it goes to the level of burning of proteins. So, this is the condition. Proteins like fats itself has low oxygen within them. So, that is why the molecules of oxygen consumed to break down this is also low, same like fats. So, that is why the RQ value is less than 1, it reaches up to 0.8 to 0.9, that will be the value. Okay, my friends, you have understood this. What is RQ? What are all the respiratory substrates you can get inside respiration? Based on the value, you can start predicting the respiratory substrate. If it is more than 1, it is organic acid. If it is 1, it is carbohydrate. If it is 0.8, it is proteins. If it is 0.7, it is fats. So, we discussed in what and all conditions these respiratory substrates will be oxidized. I think if you are, thinking about it a lot, if you have understood the entire chapter completely, entire concept of RQ, I will raise that curious contest question to you. Before that, we do previous class contest question. Okay, we haven't seen anybody coming with the right answer. That's okay. We'll discuss what is the answer of this is in ETC cycle, that's nothing but your electron transport chain, the final acceptor of electrons. Okay, I'll put it here. This is how we saw the cytochrome complex 1, 2, 3, 4. At the end, the one which accepted electrons was your oxygen molecule. It accepted 4 electrons, 4 protons. This is what we discussed, isn't it? It forms 2 molecules of water. Okay, so what is the one which is finally accepting the electrons? See, is your oxygen, isn't it? So, the answer for this contest question is oxygen, C. Let's get into our contest question, kids. The RQ value for anaerobic respiration is what? We talked about aerobic respiration only so much of time till now actually. So, what do you think is the RQ value of anaerobic respiration? Think. Come up with the comments, please. How many of you are seeing me? Tell me at least hi through comment. Yes, kids. Anything divided by 0 is what? Is your hint. Think. Yes, kids. Come on. Okay, 5, 4, 3, 2, 1. It was a snapped up countdown. So, we are done. We'll go to the next discussion. ATP yield in aerobic respiration. Okay. So, what I have done here is all the steps which we learned in a previous class: glycolysis, link reaction, Krebs cycle, we studied about ETC also. Now as I told you, as I started off the class and mid of the classes also, keep a keen note on which are all the steps where your ATP is released or your reduced coenzyme is released, okay, or which is the step where ATP from GTP also was released. So this is what I was asking you to do, because at the climax stage to rate our aerobic respiration, it would be much more useful, okay. So this is what you see is glycolysis, the first common step of anaerobic aerobic respiration. Glycolysis, as it takes place in cytoplasm, it was called as EMP pathway. We told why it is called. The German botanist, the German scientist who actually discovered this, okay. So this reaction which I am focusing down here is payoff phase. We studied about preparatory phase and payoff phase, isn't it? So why have I picked off payoff phase? As the name suggests, this is the steps of reaction where something is getting paid off, okay. So you note down here the output of glycolysis after every step or every reaction we did that, okay. Let me write it much more neatly. What color shall we use? Okay, let's use this purple color. So the output of glycolysis is, yes my friends, you see here 2 molecules of reduced coenzyme, so that will be 2 molecules of NADH plus H plus. We will go slowly, we have some time for this, okay. We will go slowly so that you understand completely how so many molecules of ATP is released, okay. And we have 4 molecules of ATP here. Why am I writing far away? Is because, as I told in glycolysis class, 4 molecules of ATP released, 2 is getting utilized in preparatory phase. 4 minus 2 will be 2. So 2 molecules of ATP is formed. 2 molecules of NADH and 2 molecules of ATP, that's what is the output of our glycolysis. So this ATP, it is formed by substrate level phosphorylation. It's happening at the substrate level itself, okay. Good, Jyotsna, nice to see you, okay. Good, I hope you have been understanding everything. Any problem, come up through comments please, okay. So this is the output of glycolysis. Let's get into the next step, link reaction. Let me use the other color here, we can use a darker green color. This was called as link reaction or gateway step. It is also called as pyruvate oxidation, okay. Why it is called as pyruvate oxidation? Because pyruvate is undergoing what? Oxidation. That's where there is NADH molecule formed. This is also called as oxidative decarboxylation, because there is oxidation taking place. This is the step where carbon dioxide is also removed out. I am keeping on repeating this because there are many important NEET questions come onto this link reaction. As I keep saying, which is the reaction which has undergone oxidative decarboxylation, which is the one which has undergone, where there is release of 2 molecules of carbon dioxide. What is happening to pyruvate after glycolysis? This and all question comes in, okay. So that is why this reaction is of higher importance in terms of your medical or any entrance exam, okay. So one more thing which we noted down here is there were 6 factors which are involved here, okay. And anybody put that out? Okay, I'll just read it out. I'm not going to write it, but it is very important for you to understand. They are lipoic acid, FAD, NAD, coenzyme A, TPP, tyrophosphate, it's called as TPP, okay, thymine pyrophosphate, Next one is Mg2+. These are the 6 factors required for this reaction. This is the first reaction of respiration where there is oxidation and decarboxylation taking place. Okay, please note that, keep it inside your brain. So what is the output of our link reaction? So this is in terms of one molecule of pyruvate we are talking. So if it is 2 molecules of pyruvate, there will be 2 molecules of NADH, 2 molecules of carbon dioxide, 2 molecules of acetyl-CoA coming out as the output. So output here is going to be 2 molecules of NADH. Triple H plus also you can put. Okay. Okay kids, the output of glycolysis is 2 molecules of NADH and 2 molecules of ATP. Link reaction is 2 molecules of NADH. Coming to the mega step, Krebs cycle. What is going to be the output here? Here we have 2 types of reduced coenzyme noted, that is NADH and FADH2. We had a step, you can note that. Again, this is like a revision class, okay? We'll keep repeating things. If something you haven't understood, common thing which I am going to say, come up through the comments. We are here to help you out. So which is that step of, I think I should use another color. Okay. Which is that step of Krebs cycle where GTP is produced? Well, there is substrate level phosphorylation. We also said that this GTP is in turn coupled to release ATP. Yes, my friends. So GTP is used by ADP, inorganic phosphate. It's going to form off your ATP plus GDP, which is getting back inside this cycle. So let us put down what is the output of a Krebs cycle. See, we always talked in terms of one molecule of pyruvic acid getting inside complete oxidation through Krebs cycle. So here we will talk in terms of 2 molecules of pyruvic acid getting inside Krebs cycle. What it will be? See, there is 1, 2, 3. 3 molecules of NADH for 1 molecule of pyruvic acid. If it is 2, how much it will be? Okay, I am putting it right here. Be 6 molecules of NADH plus H plus. Okay, there is only 1 molecule of FADH if you see here. I put in blue color, not in blue color. Let me use the other color. There is too much of colors here. Keep noting down where I am tracking off to write it. Okay, so it is 2 molecules of FADH. 2. Yes, kids. It was 1 molecule of FADH for 1 molecule of pyruvate. It will be 2 molecule of FADH2 and 2 molecules of ATP. Yes, there is only 1 molecule of ATP for 1 pyruvate or 1 acetyl-CoA. It will be 2 molecules of ATP. Right. Let's come into the complete conclusion. How much we have got? The NADH 6 in Krebs cycle, 2 in pyruvate oxidation, 2 in NADH. That becomes 10. Okay. Let me write it down here itself. It will be 10 molecules of NADH plus H plus. Yes, kids. Okay. There is no molecules of FADH formed before. So it will be 2 molecules of FADH2 formed overall at the end of complete oxidation of pyruvate, and we have 2 molecules of ATP in Krebs cycle, 2 in glycolysis. So that will be 4 molecules of ATP. Now you see, next step pasted off the same picture which we learnt in ETC. So if you have any problem, okay, to go through this, I will once again revise it off. So this is complex 1 which has FMN as prosthetic group. Okay. So the question where you went wrong, the CPP question where there are BC1 complexes, you see that. So this is what I was talking about. This is complex 1. Complex 2 is your FADH where it gets entered into complex 2 is succinate dehydrogenase. Okay. Complex 3 is your QH2 cytochrome C reductase. So this is the complex where there is cytochrome B and cytochrome C as components. Okay. Which is the cytochrome molecule? You can, anybody can raise the question is, which is the cytochrome molecule where there is AA3 composed of, where there is presence of cytochrome A and A3. is cytochrome C oxidase. Your contest question, okay, which is the one which accepts the electrons at the end of ETC? The final electron acceptor is your oxygen molecule. That is why this entire ETC is also called as terminal oxidation step. Okay, terminal oxidation process. Terminal at the sense at the end. Oxidation means this is an oxidative process. Okay, so that's why it's also called as terminal oxidation step or process. Yes, my friends, we are here at the ATP yield. Now, based on this, we are going to rate out respiration. Okay, so overall, we just now we wrote that. Okay, there is 10 molecules of NADH form at the end of respiration completely after Krebs cycle. 2 molecules of FADH2 formed. Okay, let me just write it off here again so that we don't get confused. Okay, at substrate level there is 4 molecules of ATP formed. Okay, so a bit of extra math. We have to do this so that we come up to say that so many molecules of ATP is out. Please keep a keen observation on this. I am going to tell you 2 such scenarios in 2 such ways how ATP can be produced out. Okay, in the sense that there are 2 conclusions that this much can be produced, this much also can be produced. So through our ETC cycle, 1 molecule of NADH, okay kids, can produce out 3 molecules of ATP. This is what we wrote in the previous class also. 1 molecule of NADH to 3 molecules of ATP it will give. 1 molecule of FADH gives out 2 molecules of ATP. This is through oxidative phosphorylation. Yes, let me use the same color now. Okay, this was the color. So we said 10 molecules of NADH, 2 molecules of FADH2, isn't it? If one is giving out 3, how many 10 is giving? 30. Let's make it much more colorful so that you remember, it helps you remembering. Oh, it's the same green color. Let's use the red color. Okay, so it will be overall 30 molecules of ATP from 10 molecules of NADH, and now it is 2 molecules of FADH to form. 2 into 2 will be 4. 4 molecules of ATP. Now overall chain level ATP means, now in our substrate level we have 4, 2. Now till here it is 34 molecules of ATP, yes, plus again 4 molecules of ATP liberated by substrate level phosphorylation. Yes kids, it is 38 molecules of ATP. Let me write 8 properly. Okay, it is 38 molecules of ATP overall. So chain level, this is the first confirmation. There is one more, in most of the textbooks also say 36 molecules of ATP. How is it? So one way it will say 36 or 38 also. In our textbook it is 36. Okay, there is no proper range like, oh no, only 38, oh no, only 36. It can range up to from 36 to 38 molecules of ATP. We just now calculated how 38 molecules of ATP. Okay, now I am going to tell you how is it 36 molecules of ATP, the second criterion. Okay, so in the textbooks, in most of the books, when you research for this, it says that, see, in this 10 molecules of NADH2, I'll use the eraser. I erased off 1 here. What did I do? Okay, in your 10 molecules of NADH, okay, how many molecules of NADH are coming from glycolysis? It is actually 2. Okay, 2 are coming from, keep a keen note on this. I hope you are understanding this. 2 from glycolysis it's coming. 8 is actually from Krebs cycle, okay, and your link reaction. Okay. So how this 36 molecules of ATP is coming out is, it is a condition. In your question, if they ask, calculate out how many molecules of ATP is completely out, you can give it as 38. If they say as condition with conversion of 2 NADH molecule of glycolysis into FADH2, note this, okay. So this 2 molecules of NADH has the tendency to getting it converted into FADH, okay. 2 molecules of FADH conversion, okay, this is the second criterion. So totally in this case the FADH2 becomes 4, okay, and your total NADH becomes 8, okay. Now let's use the next slide. I think there is no space here to write out. So for now we are going to calculate with 8 NADH and 4 FADH2 and 4 substrate level of phosphorylation. I'll do this side, okay. So if 8 molecule of NADH is produced out, 8 into 3 is how much? 24. 8 3s are 24. Now our FADH2 body has become 4 molecules of FADH2. Each molecule of FADH produces 2 molecules of ATP. So 4 2s are 8, right? We said 4 ATP from substrate level. Yes, see, I'll put this in the previous slide as the first criterion. I'll put in the same this one. Whatever you see, this is the first criterion. Okay kids, understood this? It's not that complicated. Understand the concept behind this. We are talking about the second criterion here. Okay, so this is 4. Let me use the same color. Let me do the justice. It is 4. Okay, so it becomes 4 and 4 8. 8 and 8 is 16. Here comes 36 molecules of ATP. Okay, 2 such criterions. How 38 if NADH is not converted into FADH2? How 36 if NADH of glycolysis is converted into FADH2? We have understood both the things here. Okay, any confusion in understanding this is much more simple. Okay, so let us give a justice, let us give a conclusion with the overall equation of respiration. Okay, we said a molecule of glycolysis C6H12O6 in the presence of 6 molecules of oxygen gives out 6 molecules of carbon dioxide, 6 molecules of water, and comes energy. Okay, so we had discussed that how 6 molecules of carbon dioxide is coming out through Krebs cycle and our link reaction, K plus L reaction. Now how 6 molecules of oxygen? Ma'am, you never told us. Oxygen is coming inside terminal oxidation or ETC to accept the electron. But how 6 molecule of oxygen? Now let us talk this in terms of first criterion. We said that there is 10 molecules of NADH, okay kids, and 2 molecules of FADH2. So this will be 12 coenzymes. Each coenzyme takes 1 oxygen atom. Oxygen is a diatomic molecule. There are 2 atoms in it. Okay, so 12 molecule of coenzyme will take 6 molecules of oxygen, as 1 coenzyme takes 1 atom. Here oxygen is diatomic. Atomic. Okay, if oxygen is diatomic, each coenzyme will take 1 1 1. Okay, so that will be 6 oxygen, 6 molecules of oxygen, isn't it? We gave justice to the entire equation. Here we have considered only the first criterion. Okay, kids. So we are done the entire conclusion. So how much would you rate respiration? 36 to 38 molecules of ATP. Not bad at all from one glucose molecule. Summary, we studied about respiratory quotient, respiratory balance sheet. We are done like a chartered accountant. We sat like an accountant and said the cell is going under credit mode, not debit mode. That is, on credit mode, from one molecule of glucose, 36 to 38 molecules of ATP. We also told how 36, how 38 also. Yes, we are coming up to we actually came up to the climax of the class, climax of the chapter. 10 CPP questions for our passport users. Answered those quickly with all the hints and whatever I've talked in this class. Okay. Any problem in answering that? Come back to us to comments. We'll try to help you out. This is a simpler chapter. It is much fun-based chapter. Understand this. NEET perspective, very important. Okay, kids. See you in the next class. Okay, have a nice day ahead. Good luck.